And we're live. Oh, hi, Sister Maya. This is our final group meeting. And for lesson 13 problems. So let's have Molly start this off. Holly, sorry. Oh my goodness, Holly, start this off.
Hey, you're totally fine. It's the same thing. No worries. Okay, so for question 1, they give you a primer sequence essentially, and they want you to, for A, they want you to show what the banding pattern would look like. So this is what I got for part 1. And then, or for part A of 1. And then for parts B through D, they want you to determine if the sequences of the other 3 template DNA molecules would, or there's mistakes, and they want to know if the reaction would fail or if it would still work. And so for part B, we got that because the DNA would denature, that it would fail because it couldn't survive, essentially. And then for part C, the DNA helicase wouldn't help or harm the sequencing, and so therefore it would not be affected. It would still work. And then for part D, it says that they forgot to add the DDGTP, the DDTTP, and the DDCTP. And so the only one that they did have was the DDATP. So on the jail, that's the only one that would show up. So it would mess up the rest of it. So their reaction would fail.
Cool. So for part 2, question 2, it's filling the blanks. And then for A, it says during a typical PCR cycle, the DNA synthesis step is done at 72 degrees Celsius, while the primer annealing step is done at 55 degrees Celsius. For B, AMPR is a commonly selectable marker that is found in many plasmid vectors, while restriction enzymes recognize and cleave DNA molecules at palindromic sites. C, during agarose gel electrophoresis, DNA is placed in an electrical field. DNA migrates towards the positive electrode during electrophoresis. For D, DDATP lacks the 2' OH group and the 3' OH group. For E, transformation is a term used to describe the process where bacteria take up DNA molecules from the environment. F, during blue light scattering, IPTG is commonly used to induce expression of the lac operon, while xgal is cleaved by beta-galactosidase to turn E. coli cells blue.
Great. And then for question 3, it says that you're interested in using the PCR to amplify the region of a double-stranded DNA fragment. And then they give you that the sequence is copied in the location between the sequences in bold below. So they want you to write a single-stranded DNA sequence that would use to, or for both the primers. And so I guess opposed to reading that all out, this is what I got for A here, and then what 1 and what 2 would be. And I'll leave that for a second. And then question, or part 2, B, wanted to know the temperature that would be used at the PCR, and they give you an equation, and so you just plug in the numbers and everything from the problem above. And so For the first primer, I guess we got 62.3 degrees Celsius. And for the second primer, we got 59.8. And then question 3 wants to know if each PCR cycle contains a DNA sequence step. What are the sequences of the 2 newly synthesized strands of DDCTP? Is if it's accidentally included in the experiment, and they say that assume that the rest of the components are found in the reaction mixture. And so for part C, for the first one, I will again just show for part C, the first one, and then the second one, right there, 1 and 2. And then for part D, they want to know, when we learned about DNA replication, we learned about DNA-A and the DAM protein, and they wanted to know why they're not included in the PCR experiment. And this is basically because they aren't involved, or they are involved in the initiation step of replication in E. coli. And so the DNA-A is going to catalyze the initial separation of both of the strands, and the other one is going to start it, the DAM protein. And so therefore the PCR cycle is going to include denaturing, and so because of this, they're not necessary. We don't need them.
Okay, and for question 4, it says this question concerns Arago's gel electrophoresis and restriction mapping. Suppose you set up a restriction enzyme, digestion reactions. Each reaction contains the same linear DNA molecule, but in each case, the DNA molecule is digested with different combinations of the restriction enzymes, high, E. coli, and HIN3. These reactions produce the following results, and then they give you the little table. So then you get, after you draw everything out, you get this little wonderful drawing. I'm so bad at drawing.
It's good.
So you get this drawing, and then just so I can quickly explain it, you have the negative on top and then the positive on the bottom. And then you have the restriction enzymes. I just labeled them from 1 through 6 on the top. And you have 100, 200, 300, 400, 500, and then it jumps up to 1,000. So then for number 1, for band 1, you have a band at 500, at 350, and at 300. For line 2, you have a band at, I think it's, let's see, 700 and 200. For band 3, you have one at 600 and 300. For band 4, you have one at 500, 200, 150, and 50. For line 5, you have a band at 500, 250, 150. And for line 6, you have a band at 600 and at 200 and 100. And then for part B is asking you for a linear restriction map that indicates the location of the restriction enzyme. So what we did was, this is our restriction map. And so we have 50, 150, 100, 100, and 500. And then a 50 BEM high, HI, whatever you call that, that's off there. At 150 is the E. coli. At 100 is the HIN3 and the BAM2. And then you have the 500 left. And for C, it says what are the predicted fragment lengths? And it's just the same thing, which is 50, 100, 150, 100, and so then, okay, sorry. You good on 5?
My computer just froze. Can you hear me?
Yeah.
I can't switch screens. Well, I try and pull it up. Do you want to start it?
Sure, I could start it. So it says you're interested in studying a gene called your favorite gene that you have identified in the yeast Saccharomyces cerevisiae. I cannot say that. It says detail the steps that you would use to generate cDNA from Saccharomyces mature mRNA, clone the cDNA version of YFG into a plasmid vector, and use the blue-white screening to identify plasmids that contain the YFG cDNA. So then for that one, for step 1, we got that you would run PCR to produce more copies. And while that's being run, that includes the DNA training process that will occur at the 95 degrees Celsius. The annealing process occurs at 52 degrees Celsius, and the extension occurs at 72 degrees Celsius. Then the second step that you would do is that you would run the agarose gel to check if the PCR was successful. And then after that, we would take the restriction enzymes and use them to cut the vectors and insert the DNA. Following that, we transfer them onto the plates. Step 5, you use the XGAL and the ITPG to see if blue or white colonies are formed. And then finally, if the blue colonies are formed, then you know that the PCR was not successful. And if white colonies form, then we know that it was. And that was our lesson 13 group meeting.